think everyone's back. Omar, is there any other comments that you want to make, or is it me?
No, I don't. Yeah, I don't think so, unless there are any questions from the breakout rooms that required sort of general discussion. People are good?
I think so. Not from us.
Okay, perfect. So I think the next section is Sam, the last part of the workshop, and that is demoing the MapVEu functionality. So go ahead, Sam, all yours.
Thank you. Hello, everyone, or good afternoon or good morning. It's very much afternoon for me. So I'm going to show you a tool called MapVEu, which is, and I'm going to do it in much faster time than the allotted block. But it's currently only available in vector-based, but we hope to soon have it available in other component sites. And if you have particularly good data sets that you think would look good, please let us know when I show, and email the help desk, and maybe we'll get it implemented in other sites. So let me share my screen here. Okay, so now this is going to be a little bit different. We're going to try this where I'm going to give some general click here or click one of these buttons, anyone, and you'll see, and so you can try to follow along. This is a kind of a follow along introduction. So the tool that I'll be showing is, you can find it, you have to go to vector-based. And remember, if you're not at vector-based yet, you can go to the bottom of any of the websites and click on the little green mosquito. And it'll take you here, and then go Tools, MapVEu. And that will take you to a page that looks like this. And as you can see, we have this map here. This is a very different interface from what we've been looking at the rest of this workshop. And I would like you to just click on a circle. And now it is kind of sometimes hard to do this because you have to like minimize your window perhaps and shove zoom over to one side. And sometimes it keeps trying to pop up and take over your screen, but hopefully you can move me to one side and we can just click on some things together. So I'm going to click on a circle, any circle. What you'll notice is that this pie chart pops up and it shows us that 45% of the samples were metadata only, 12% were abundance for mine. So take a look at yours, see what you see. You'll see that we have the search box up here. We have our left hand menu items, and we have what I call our right hand drawer. So these are kind of the parts of the screen that I'm going to talk about. So yeah, now, so click on a point, see what happens. Then I want you to double click on a point and see what happens. You'll notice that wherever you clicked on, it should zoom in, and you can probably keep clicking on points and zooming in. So try that, and you can keep doing that until you get to one that has this little picture of a pin here. And that means that you can click on it no more. Well, it might zoom in more, but it's not going to disaggregate into further points. So this is called an atomic point, and it's the most resolved points that we have. Now, in this view, you just see that there's some kind of data that's at a geographic location. So I'm going to click this world here and zoom back out. If you want to do that, you can click on the world point, the plus point, the minus point to zoom in and zoom out. But it's much more powerful when you start looking at particular data types. So right here, you can pick what we call views. So I'm going to click on insecticide resistance. And you can do that. And then now you see a different view. So a lot of the data points went away. So now we only have data here that is insecticide resistance. So again, click on a point, any point, see what happens. And we still have this pie chart, but now there's some more buttons here that we can go through. And some of these will not work depending on what view you're in. And what we see here, when I click on this second button, so click on any point and then click on the second button, and you can see the data that underlies that we have from that point. And we record all the metadata, just like you've seen in other parts of the site. So you can highlight one of these points, and you can see exactly the underlying phenotypic data from a assay was collected from a mosquito in the field that has this insecticide resistance assay performed on it. So we see the date that it was collected, where it was collected, the method of collection, which insecticide, which percentage, and how long it was exposed, and what testing protocol. So in the one that I have, WHO testing kit, but you might see a CDC test. You might see, instead of lambda cytherin, you might see permethrin, you might see deltamethrin, you might see some more exotic insecticide. So try that, click on anything and see what kind of insecticide data you get. You can also maybe filter. So maybe you only want to see one species. So go to the right-hand drawer and you click on a species, and you'll notice that whichever one you clicked on will appear here in the search bar. And that means that and more points have disappeared. So we just have points now that have data from Aedes aegypti. And by changing the drop-down box, so I hope you're following along, just click on any points and just generally do what I do. You don't have to try to recreate the exact things that I'm clicking on. So right now I just have Aedes aegypti, but I can also say I just want to see studies done on deltamethrin, for example. So now I filtered out more things. I'm only seeing assays that we have on Aedes aegypti and deltamethrin. And you can keep adding as many of these as you want. You can also do this instead of clicking through here. You can just type in a species name, for example, or any of the studies that any of the metadata terms you can also search for. So I'm going to type Anopheles gambi, and so now more points are going to appear. Now, just as a little reference, in our tool, if you have like two species, that will be an or, if you're familiar with Boolean. So this will be Aedes aegypti or Anopheles gambi that does use the assay use deltamethrin. So, and again, we can always click and see the graph from the data at that point. And if we double click and zoom in, it's going to disaggregate, and that graph will change to represent the data that we have on the screen. You can see all the data from that point in tabular view, no matter where you are. You can always download the data, and it's going to be all the metadata that you need, that you saw in the tab view and more will be available here. In fact, if I click here, you can see all the fields that will be in the resulting data table. And a really cool view is, let's say, you have this search and you found something really interesting and you want to send it to your PI, your colleague, etc. We'll zoom out so we get a more interesting view. You can always click the share button, and that'll generate a link that you can copy and paste, put in an email, and then when you send it to someone and they click on it, they can see approximately the same view. So I've shown you insecticide resistance. So now let's go to abundance, for example. So this is a very different kind of data. This is spatiotemporal repeated abundance data. For example, a lot of, maybe you're a mosquito abatement district, or you're doing a long-term study where you're collecting mosquitoes every week or every month. So let's change that view. And I'm just going to get rid of all our filters, and we'll see we have lots and lots of data. And I'm going to zoom in here to the United States. I'll just pick, I don't know, somewhere in Florida. We have lots of data from Florida. And I'll click on a point. I'm going to zoom in a lot because there's a lot of data here. And we always have a pie chart, but now when we go back here to this left screen, it's not, you would expect that any resulting graph is not going to look like the previous one, because it's not insecticide resistant. It's a completely kind of different kind of data. So when I click on that, I see that this kind of data is going to instead look like a time bar, the change over time. And I can change the resolution. And this looks kind of boring. So whenever everything is gray, we use some algorithms that calculate colors, but when there's too many species, it doesn't, you need to tell it to retry. Let's click optimize colors, and then it'll recolor everything, and I can see the resulting graph. And just like any other view, we can always filter. And this time, instead of a particular species, I'm going to say a particular genus. And then it will recalculate, and I'll change it back to, so we have yearly, monthly, epiweekly, sometimes even daily. I'll change it to epiweekly. Apparently there's a gap in data that maybe they didn't collect from that location, so I'll just do that. Now I have a graph of the mosquito populations over time from this location. We have genotype view, which right now is all insecticide resistant genotype instead of phenotypes, where you can look at the percentage of genes of particular alleles at different loci. A pathogen view, which shows pathogen detections over time. For example, some places do long-term surveillance where they're not only counting the mosquitoes, but they are testing them for presence of pathogens such as a virus, arbovirus, or malaria. And blood meal analysis, where they assay the what the source of the blood meal was, whether a human, a cow, a bird. So a very large epidemiological importance. And all these tools work the same. All these features will be here. The graph might look differently depending on if it makes sense for that kind of data. You can always filter either using the search bar or using the right-hand drawer. You can even, for some of these, like abundance, you can do which attractant. Maybe I only want to see things that were attracted by CO2 or only attracted by light, or I want to only see things. So here I show attraction, but you might also search by like trapping type. So sometimes, depending on which trapping type and which attractant you use, you can really are biased towards one kind of mosquito vector or another. I know that was really fast, and I hope you've just played around and clicked around. Unlike some of the strategy systems where one errant number will give you maybe a spreadsheet that doesn't make any sense. This is very much just a good way to discover where data is, and just feel free to click around. I hope you see the value if you have any spatiotemporal data. And like I said at the start, this is just available in Vectorbase, but we are very much looking forward to data sets that will work well in this system for some of our other sites. Are there any quick questions before I let you guys go? Let me see here.
That's great, Sam. That was a super fast way, so I'm sure people will be rewatching the video as well. But very nice, quick demo of all the features that are available.
I'm going to see if there's a taster.
Yes.
The trailer. It was the trailer version.
Great.
Okay.
Well, so we are, I guess I'll share my screen as well. Okay, and back here. Okay, so we are at the end of the workshop. So we don't have any more exercises for you to do today. So I think we'll hang on for another 15 minutes or so to answer any general questions you might have. I believe we'll try and send you a questionnaire maybe next week sometime. It'll just be a generic, what did you think about the workshop, and let us know if you have any suggestions for what to do. And we would appreciate, obviously, any open-ended responses you give us for the workshop, both to improve it and also what things you thought worked well and you would like to see more of. So that would be appreciated. And Suzanne will probably send something out either later today or sometime next week. It's not a big deal. No rush on that, obviously. So yeah, so if there are any questions from anybody from the attendees or any of the instructors, any additional comments you want to make?
There's a question about certificates, and we will send an email to folks who have been here at Zoom. And it'll include a form that when you fill out the form, it'll automatically generate a certificate. So look for that email. Yeah.
Request for a group photo.
we can do a group picture. We don't have everybody who was here on the first day or the second day, but that's fine. If people would like, feel free to turn on your cameras and we can do a group picture. Oh
Maybe stop sharing.
yeah. Stop sharing. There we go.
Yeah.